alpha-glutamate. You follow? So this one becomes alpha-glutamate, and then the byproduct is acetoacetyl. So some of the biochemists will say, you added this 1 month, of course it will come out as glutamate. It will accept the amino group here, because this cannot undergo transamination. Diba? You have to remove the amino group. And there's actually other amino group pa in lysine. Okay? So for some, they said this is purely ketogenic, because you added this one, it come out like this, then it doesn't count. But other will say it should be both glucogenic, because this can become alpha-ketoglutarate and then gluconeogenesis. Good job. I hope you know how to appreciate that one. Okay? A nicer version of what you have in your book. Yeah, another one, we have one saying the same thing. So let's go to the product. So the derived amino acid, which is also important, will come after translation has happened, post-translation. Okay? So I want you to remember this one. We will be talking about this when we go to vitamin K and when we go to the coagulation system. So carboxylation of glutamate, it even has a 3-letter amino acid form, GLA. So GLU, glutamate, GLN, glutamine, GLA, carboxyglutamate. It will give you a lot of negative charges which can interact with the positive calcium. Then you know about hydroxylation of proline, hydroxylation of lysine to eventually form a very stabilized collagen fibers. Okay? So let's take a look. What is this? Dopamine, norepinephrine, epinephrine. These are your catecholamines, so neurotransmitters. Okay? It comes from, what is that? The same as in hydroxylation of your phenylalanine. Okay. So it becomes dopa, you remove the carbon, become dopamine. And then you remove the water, become norepinephrine, and then you add the methyl group, it becomes norepinephrine. So you have the methyl group here. So if I ask, give now, these are the 2 amino acids that you need to form epinephrine. The answer is tyrosine and methionine. Okay? But to form dopamine and norepinephrine, tyrosine lang. Okay. Again, tyrosine, melanin. Okay, so your skin pigment comes from tyrosine. Then what is this trimethyl lysine? You have 3 methyl group at the last end of your lysine. It can become your carnitine. And what is carnitine? What is carnitine? Beta oxidation will not happen if you do not bring your activated SL-OA into the mitochondria. Okay. Tryptophan, here we are, as niacin, NAD, as melatonin, and as serotonin. So a methyl group is added. So it can come from some. So from tryptophan, you have, what is the other name of your serotonin? It is 5-hydroxytryptamine. You hydroxylate and then you decarboxylate. Okay? So 5-hydroxytryptamine. Okay, creatine, very simple. You need lysine, you need methionine. What else do you need? Arginine. So 3, arginine, lysine, and methionine. Become creatine. Where are we talking about creatine? Because of this one. Okay? Arginine gihapon, methionine to become your spermidine, spermine. Why are they important? They are polyamines. Okay? To stabilize the DNA molecule. And I did not explain this anymore. What is lacking here for the purine? Okay, glycine. This is in your book, but the book actually said that the carnosine, which has histidine, okay, and homocarnosine are major constituents of excitable tissue, the brain and the skeletal muscle. So they know they are there, but they are not sure what is their function. Okay? Maybe if you become a researcher, you may be able to elucidate the function. Okay, this is about serine and glycine. Cysteine, you know already this one? Can become your CoA. So there is part of cysteine in CoA. Then you have your serine in your sphingosine to form your sphingolipids, right? There, the phosphatidyl-choline. So glycine, purine, bile salts, your porphyrin. What has to be added to form your heme? Glycine plus succinyl-CoA is the starting point to form your heme. The compound product will just keep on condensing. Okay? Of course, your glutathione, your 3-tripeptide. Okay, a lot. Glycine can also be used as a conjugating compound, okay, to take care of your. And this is the last. Very small lang dioecium, NO lang. But it has a lot of functions. Short-lived signal molecule with diverse role in different cell types. It comes from arginine, and then take note, you read your NADP, to convert arginine to nitric oxide. See? NADP. What are the functions? Vasodilatation, regulation of smooth muscle contraction, gene transcription, kopya na lang po anak. Neurotransmission in the CNS, regulation of penile erection, non-specific host defense mechanism, the macrophage healing, inhibit the proliferation of your inhibit the proliferation of tumor cells and microorganisms, induce apoptosis. Okay? Actually, your NO is also carried by your hemoglobin. Okay. This is an assignment that I will ask the group to write and submit, because I'm not going to interact with you anymore about hemoglobin. Huh? Hemoglobin is part of the respiratory system, and that will be taken up when we are talking about transport of oxygen.
So for tomorrow.
So you read what is in Harper, and you read some more what is in the belief, because the role of the NO is only found in the belief. NO, 50%. So not only is the hemoglobin able to bring in the oxygen, it will make sure that it will receive it by vasodilating.
Side by side sila. Because most of the time, I don't really totally understand this.
So
I understand Devlin is nicer. It's student friendly. And the pathways, yeah, the pathways dyan kasi parang ganun, something In Dublin, it's like, ano ba? They break it. So you see, ito. That's how I understood.
What about? The one below?
Because it's
You will
longer
talk about the
than that.
hip packet. You will talk about the T and the R form. And you will talk about the NO. You get me? Hip, globin, function. Then below is the hip packet. It's important. And then the T and R differentiation. And then the NO. And then the positive operativity capacity.
So, kasama yan tomorrow.
So the positive operativity is 2 for loading oxygen.
Yeah, I
and
think it's
unloading
kasama
oxygen
tomorrow.
in the hemoglobin molecules. Okay, any questions?
No.
You will pass that now. Okay, you will be very happy. I will not include this in the unit exam. I will include this in the bi-monthly exam. That's why I want you to make it. You will pass this on Tuesday. Tuesday, Monday, there is no class. Tomorrow, you will have your research lecture on study design. Okay, different kind of research studies. Again, with